Hey guys, welcome to topic 2 of chapter 9. In this topic, we're going to talk all about the tree of life. So in topic 1, we talked about genetic variation. In topic 2, we're going to talk about how this genetic variation across all species has led to the tree of life. So in this topic, we're going to talk about the tree of life, selective advantage, conserved sequences, evolution, and we're going to then focus a little bit more on the human genome and what we do know, and more importantly, all the things we don't know. So we've been talking about this a lot, and this hopefully wraps up a lot of what we've been talking about this entire unit. So here are your topic objectives. As always, if you have any questions about them or need any help with anything, please let me know. So let's talk about what we're going to talk about in the tree of life. When we're focusing on the tree of life, we're going to talk about this role of selective advantage. We're going to talk about comparing the genomes between human and chimpanzees, conserved islands of DNA, comparing the genomes between vertebrates, and then drawing the tree of life. So here's our tree of life. First of all, you've seen images like this a lot. We currently are operating under the 3-domain system. If you were in high school any certain number of years ago, you may have learned about the 5-kingdom model, and that's been changed to 3 domains, which consists of bacteria, archaea, and eukarya, with bacteria and eukarya being at the far ends of the tree and archaea being in the middle, symbolizing that it broke off. That bacteria broke off first, and archaea, and then eukaryotes as far as in relation to us. And this is mostly due to sequencing techniques that have developed over the last several decades. Homologous genes are used here to help us figure out what's similar between them and what's different. And so that's how we were using this, is we can compare and contrast to see how similar things are. So remember, back when they first started sequencing everything, they went off of what they looked like, where they lived, what they ate, and now we can see how similar they are to each other in that way, and that's actually how we can move things around. A lot of the big shifts have happened in the bacteria domain, but there's also some other shifts that are occurring in other places. But as far as we're concerned, we're just looking at the big overview. This is not a phylogenetic class. So let's talk about selective pressures. We've talked about this before, that evolutionists change over time. So you have to have the ability to change, which we talked about with genetic diversity in the last the last topic. And now we're going to talk about how that change over time drives it, and that's through selective pressures. Now remember, I could contain the most awesome genes in the whole universe, but if I got hit by a bus tomorrow, it wouldn't matter, because my genes, number one, wouldn't be passed on, and they may not even be expressed. The key to selective pressures, especially with regard to the tree of life, is not only just being able to reproduce, but reproduce successfully. So it's not even that my offspring survived and have my genes to pass on. It's that they then continue to pass on those genes. And if we're going to talk about evolutionary success, the more you pass on your genes, the more successful in an evolutionary sense you're talking, as opposed to organisms that either have only one offspring that may or may not live to adulthood to reproduce. So it's really important you understand that it doesn't matter how great the genes are if they can't be passed on. But if they're great genes, they will hopefully help you survive to pass them on, and then hopefully help your offspring pass them on, etc. Now what you can see here on this slide is we have an image of how the last common ancestor of all primates is at the top of this pyramid shape, this triangle shape here. And you can see that orangutans and humans differ by about 3% of their nucleotide substitutions, whereas us and chimpanzees are about 1%. And most of these differences are neutral mutations. However, that's not always the case, because obviously if they were all neutral mutations, orangutans and us would look identical. We would be the same species. So the changes do have to impact things such as regulatory genes or even genes themselves. So it's really important to understand that just because a lot of them are neutral mutations, and not all of them are. So let's look at humans versus chimpanzees. We share about 25,000 genes, which is a lot, and most of our chromosomes are pretty similar. The big thing is that our chromosome number 2 is split into 2 in most higher order primates. And you saw that when we talked about the muntjacs, is that chromosome rearrangement happens all the time, and organisms can still be pretty similar. Another big key about humans and chimps is that a lot of our mobile genetic elements are pretty identical, which means that a lot of that movement that we saw and that we've talked about with these mobile genetic elements occurred before our last common ancestor. So it's important to understand that. But what you can see here on this slide is the few places where there are amino acid changes between the chimps and the humans. And it also shows that the gorilla changes on here too. And this shows us that we're about 1.2% difference in sequence scattered throughout our genomes. And that's what equals chimp versus human. Now, And we're going to talk about this throughout this entire lecture. We're not entirely sure what it all means. All we know is that it's different. So at this point, we have the technology to show us the differences, but we don't have the understanding and the technology to be able to tell us what all these differences mean. So this takes us to our next topic of conserved islands of DNA. So we've talked about conserved islands and conserved DNA, and what this means is it's sequences that are similar across multiple species, or if you're talking about within a same species, multiple individuals. But in this case, this entire lecture, we're talking about multiple species. So what you can see here is anywhere there's green on the mouse versus, well, anywhere there's green on the mouse is similar to the human. And you can see where those conserved are. So about 50% in the there's about 50% variation between the human and the mouse, compared to the 5 base pair differences between human and chimp. So that's showing you that there's a significant genetic difference between us and a mouse and us and a chimp. And that makes sense if you look at even our physical traits, that there would be a significant difference even in size between the human and mouse genomes versus the human and chimp genomes. And these segments without changes are likely to be results of purifying selection. And what that means is that if these changes occur in this area, it's going to cause a lethal change or something that can't be survived. They have to be there. So the purifying selection is where the genes cannot be changed no matter what. So remember that purifying selection means that if it's changed, chances are that offspring or that person or that organism is not going to survive, because it is that important to life. And so here's some more. We can see this with comparative genomics. What you can see here is the area of green is overlapping information. So starting at the top, that orange bar is the human genome, or is a human gene rather. The next bar is chimpanzees. You see how solid green that is? That's showing just how much identical that gene is between chimpanzees and humans. And as we trickle down, you see that the green gets less and less, and that's because the percent identity is changing. But those areas that stay green through a majority of the organisms are conserved islands of DNA. Those are genes that everybody must need to have for them. About a third of them code for protein products, but others code for RNA, gene regulation, things like that. And some of them we simply don't even know. All we know is that they are conserved. We don't know what they do, but they're clearly very important, because otherwise there wouldn't be as much homology between them all. And so if you look at this, most of our mammals have similar sized genomes, and reptiles and fish and bird, obviously, as we know, have split earlier in evolution. And so that's why the genome size varies so differently. Now, one interesting thing is the puffer fish, and it has 1/10 the size of our genome, and it contains almost no introns. But the introns it does contain are almost identical to ours. So it's just incredible that their genome is so small, but it matches ours so closely. And as I said, this is all just due to when they broke off and how they kept evolving into their own distinct pathways. And these breakoffs have more than just small mutations. These can come from whole genome duplications or whole gene duplications, or they could be pretty big changes. And that's where we got the variation within all the branches of the tree of life. So here we are again, the tree of life. And as I mentioned before, what we did, part of what we do now that we have the sequencing data, is we look at the small ribosome subunit and we compare how different those sequences are to each other to help determine where the breaks are in the tree of life. And as you can see here, there's a lot of similarity between the human, Methanococcus, which is an archaea, E. coli, which is a bacteria, and human, again, because we're at the bottom just to help us sandwich it. And that helps us see where these breaks would occur. So that's how we use this to do it. So we started off, and we didn't totally wipe the tree of life clean when we got this information. We just use it to refine the tree of life that Darwin and everybody else from early on started. And this just helps us refine the tree of life. So now let's look at the human genome. We're going to talk about how our genes are arranged, what makes us human, and everything else. So we've talked about this, the human genome. We have 23 pairs of chromosomes, 22 autosomes, and 2 sex chromosomes. Hopefully at this point you really have that nailed. We did this sequencing first in 2001, and the published sequence of our genome is a compilation. It's not just one person's, because it makes sense, because with the single point mutations within them, they wouldn't make, it would be hard to just have one person's genome be the standard human genome. So we did a compilation. And we found that we have about 3.2 times 10 to the ninth base pairs, and on average we differ by about one base pair per 1,000 base pairs in each other. That's how different it is on average. That I am from you, you are from somebody else. So how are our genes arranged? As we talked about this before, our genome has a sequence. We have about 5% that's highly conserved across all mammals. That means that there's a bunch that's going to be related across everything else. And a lot of this has to do with some of the sequences we've talked about, the regulatory genes, some of the mobile genetic elements, things like that. But we still don't know a whole lot about it. What we really know is that we know what it's composed of, but we don't understand what that means and how this works. So it's really important to understand that we have repeated sequences and then unique sequences. And about half of our genome is repeated sequences. So this is still a big growing area of research. And if it's something you're interested in, I highly encourage you to to explore this as an option for your research in your upper division classes or even your graduate school, because we still don't understand what a lot of this is. And as I said, we don't know what it means and how it makes us human. We have found some regions called human accelerated regions, and this is where after we broke off from chimps, we saw a huge amount of change. So for example, there's one region where there's 18 differences between us and chimps, while chimps and chickens, remember birds split off a long time ago, they only have a 2 base pair change. So that big change may be something that led to our human characteristics very different from the chimps. But as I said, we're not entirely sure what it all means. So make sure that it's really incredible to know this. And if this is something that intrigues you, I really encourage you to continue exploring this area of research because it's only speeding up. The more our technology improves, the better we are at understanding what these changes are meaning. But what makes us different from each other? So we know we have all these differences from chimps, we know we have all these differences from fish, etc. But what makes you and I different? What makes you and your cousin different? And we all differ by about 0.1%. And the variations mostly come from about 100,000 years ago, with single nucleotide polymorphisms being the bulk of these changes. So just every once in a while a base pair change. Now most of these didn't cause a big problem, so it's really important that we know that. Now remember, if these variations were purifying agents, that would mean that they would be incompatible with life and wouldn't have been passed on. So that's why most of these variations have to be silent, because in order for them to have passed on, they couldn't have been harmful changes. And these can be either duplications, deletions, repetitive sequences. So it's not a huge amount of difference between us. But this is what can lead to blue eyes versus brown eyes, blonde hair versus brown hair, height, weight, earlobes attached, hairy fingers, wet versus dry earwax. All of those things is what can lead to some of that variation we see. So as I said, in summary of Chapter 9 and the Tree of Life, especially Topic 2, we still don't know a whole lot of information. Not only have we learned all this about genes, but now we know about epigenetics, which adds a whole nother complicated layer on top of it. Because even if your genome's the same, your epigenetic code, if it's different, can cause your genome to behave differently. And we're not even sure how that even begins to be impacted and how we can modify that and how big of a change that is. So there's just so much to explore, and I really encourage you to think about this as something to continue working on as you move forward in your scientific career. So this is the end of Topic 2 for Chapter 9. If you have any questions, please let me know, and make sure, as always, that you review the objectives.